Hello friends. In the last lecture, I discussed about the morphological changes that take place in the reversibly or irreversibly injured cell. And today I will discuss if irreversibly injured cell injury has taken place. Irreversible injury is followed by the cell death. What kind of cell death are there? So I will discuss those things. So there are two kinds of this cell death. One is called as necrosis and second is called as apoptosis. So as I told, irreversibly injured cell has no other option. It is only option that is left for an irreversibly cell injured cell is that it is going to die. That is cell death. And most of the time we discuss that cell undergoes a necrotic cell death, which is a very common form of this cell death. But it is not the only kind of a cell death. There is another kind of a cell death which is called as apoptosis. So I will just discuss the key points of the necrosis and apoptosis. What are those differences? Number one, I'm starting from the apoptosis. So the key line of apoptosis is that it is a programmed cell death. Programmed means you have programmed something. That means this thing will happen, this thing will happen, this thing will happen, and you have programmed this kind. And those lines they happen in this way. So this apoptosis is a programmed cell death. But necrosis, it can happen at any time, in any manner. There is no program that cell is going to die. So this is an unprogrammed cell death. So, and when we say necrosis, it is pathological. That means it happens whenever there is a cell injury, there is a disease. But apoptosis can be physiological. That means it can happen in a normal cell as well as it can be pathological. Necrosis involves inflammation. We will discuss inflammation in detail in subsequent lectures, but in this condition there is no inflammation. Cell swelling is the first sign in the necrosis. Now in this condition cell shrinkage is the first sign. So necrosis is executed by general enzymes. Apoptosis is carried out by specialized enzymes, particularly these are called as caspases. And as I told, it is apoptosis can be physiological. Physiological means, you remember, if the child fetus is developing, so many things are dissolved. Like I'm saying, this is my hand. So if the fingers are deformed, the in between the material has to be dissolved. So this kind of the thing which is present inside the two fingers, if it is dissolved, if it is killed, it is removed, only then the fingers can be formed. Okay. So when this kind of a cell death is taking place, this is not necrosis, this is an apoptosis. So now, how we can distinguish that this cell is necrotic or it is apoptotic? So there are a number of methods. I'm just discussing one of the methods, how we can distinguish these kind of this cell death. Let's say you are having a DNA of a necrotic cell. That means take a necrotic cell, isolate the DNA. Okay. And similarly, you take out an apoptotic cell, take out the DNA. What you do? You carry out a gel electrophoresis. Gel electrophoresis is a method to separate the charged molecules. Okay, so if you carry out the DNA separation, so this kind of a pattern is produced. This pattern is called as DNA smear, and it is a feature of a necrosis. But you see what happens in the apoptotic cell. In the apoptotic, there is no smear. These kind of ladders are produced. This is called as DNA laddering. So all the DNA is not there, but one DNA is separated from another. So this kind of this is a typical feature of apoptotic cell death. This kind of a cell is a feature of a necrotic cell. This is one of the way to distinguish the kind of a cell death. So in this lecture, I will elaborate more on a necrosis, not on the apoptosis. I will discuss apoptosis in the subsequent lectures. So as I told, necrosis is the most common form of the cell death in the pathological condition. Remember, I am using a word pathological. So there is a definition of a necrosis. What is the definition of necrosis? Necrosis is defined as, this is you can say textbook definition of a necrosis. What is that definition? Sequence of morphological changes taking place in an irreversibly injured cell. This is a definition of necrosis. Why this kind of a definition is there? Because the necrosis is distinguished on the basis of morphology, how it looks like. And it happens after irreversibly injured cell. So that is why it is defined as morphological changes taking place in the irreversibly injured cell. So necrosis is not of a single type. There are many types of necrosis, like coagulative necrosis, liquefactive necrosis, gangrenous necrosis, caseous necrosis. I will discuss these necrosis one by one. First of all, I am going to discuss about the coagulative necrosis. What is the feature of coagulative necrosis? Now, the very important feature of coagulative necrosis is that if this cell is there, it has died, it has died by a necrotic coagulative method, then even if you see a cell structure will be preserved. Cell will not be very much different from a normal cell. Preservation of the cell structure is a feature of a coagulative necrosis. For certain period of time, obviously no cell can remain in the same stage if it has died for a very long period, but for a certain period of time, the cell structure will be maintained. And what is the reason of the cell death? Cell death takes place because of the denaturation of the protein, non-functionality of the proteins, whether it is a structural protein or it is a functional protein. So remember, the DNA proteins become non-functional. This is the key feature. So because the denaturation of all the proteins has taken place, so no digestion can take place, because digestion has to take place by the lysosomal enzymes. Lysosomal enzymes are also not active. If they are not active, how the cells will be digested? So it causes the denaturation of the digestive proteins. So cell is not digested. If cell is not digested, cell frame will be maintained, cell structure will be maintained. Where this kind of a cell death take place? Example is if hypoxia take place to the heart, that is in the form of the myocardial infarction. You take out the cell structure is maintained for a certain period of time. So this is called as coagulative necrosis. Okay, so now next type, liquefactive necrosis, is very different from coagulative. Now you see, in this condition, digestion of the cell take place, and that digestion take place in such a manner that there is a formation of a viscous mass. And because the cell is digested, it has become a liquid mass. Obviously, you can say there is a loss of the structural framework. So, why that digestion has taken place? Why the cell is digested? Because of the release of lysosomal enzymes. They are digesting it. Where? It can take place if the hypoxia take place to the brain in the central nervous system. So, this is the second type. Third type, gangrenous necrosis. In gangrenic necrosis, where there is a gangrene. Gangrene means there are two things which are happening. Number one, there is a ischemic injury. Number two, bacterial infection. There is two things which are happening simultaneously. So, ischemic injury is superimposed by bacterial infection. So, example, if the person is a diabetic, long-term diabetes mellitus is there, then he suffers a lot of complications. One of the things is diabetic foot. That means there is some ulcer or some problem happens in the legs, and the whole legs become black in color. That is called as the gangrene. So, in that condition, there is a hypoxia, ischemia plus bacterial infection. This gangrene can be of two types. One is called as dry gangrene. Dry means when the ischemia is predominant and infection is lesser. There is a second thing, wet gangrene, when the opposite is there, when infection is predominant and ischemia is minor. So this is a gangrenous necrosis. Third type is called as caseous necrosis. Caseous means cheesy like appearance, whitish cheesy. So whitish gross appearance of this necrotic portion is a feature of a caseous necrosis. So why this kind of a caseous necrosis? It is a mixture of two. It is a mixture of coagulative necrosis and a liquefactive necrosis. Both the things are happening simultaneously. That means the central portion where the cheese-like portion is there, it is a feature of a liquefactive necrosis, and around that cheesy portion there is a intact cell structure, that is a feature of a coagulative necrosis. So central necrotic portion like cheese, which is a liquefactive component and is surrounded by intact cell component, there is a coagulative necrosis. And where caseous necrosis take place, example, it happens if there is an infection with the mycobacteria tuberculosis. So this is what I wanted to tell about a different kind of the cell death. In the upcoming video, I will discuss about the free radicals, the kind of the oxygen reactive species, how the free radicals are formed, what are the mechanisms involved in cell injury, and what are the antioxidants. Thank you.